Greetings to one and all. It gives me immense warmth and great pleasure to grace all of your presence in the interest of everyone. I, Ms. Shelly Chauhan, Galgotias University, welcome you all on the third day of the seminar, organized by the Department of Forensic Science, School of Basic and Applied Sciences, Galgotias University. The previous session was graced with positive feedbacks by our participants, and we hope to keep up your expectations. Now, we begin with the Faculty Development Program. It will be headed by Dr. Saroj Kumar Amar, Postdoctoral Research Associate, School of Medicine, Yale University, United States. Dr. Saroj Kumar Amar holds the post of Orise Postdoc Fellow in the Environmental Laboratory at US Army Engineering Research and Development Center at Vicksburg, MS, United States. After completing his bachelor's and master's with biology and chemistry from University of Delhi and GGSIP University in Delhi, respectively, he has pursued his PhD from CSIR, Indian Institute of Toxicology Research, Lucknow. Then after he did his postdoc in phototoxicity from Yale University, USA. Also in neurotoxicity from US Army, ERDC, Vicksburg. He has worked as assistant professor at School of Bioengineering and Biosciences at Lovely Professional University, India. Also, as a postdoc fellow at Department of Therapeutic Radiology, Yale University. Till date, he is the author and co-author of 14 peer-reviewed publications and books such as Photocarcinogenesis and Photoprotection, Skin Aging and Cancer, and Current Advances in Breast Cancer Research, a Molecular Approach. Sir, I request you to kindly take over.
Thank you. Thanks a lot for my brief introduction. Am I audible?
Yes, sir, you are audible.
Just a minute, I am going to share my PPT. You able to see my screen?
Yes, sir, it's visible.
Hello, good morning, Galgotias. Many thanks to convener and dean, School of Basic Science, Basic and Applied Science, Galgotias University, Dr. Arvind Kumar Jain. Thanks to organizing secretary, Forensis Agora, Dr. Rajiv Kumar and Ms. Vinny Sharma and the entire team of Forensic Science, my dear student and colleagues. It's my pleasure that once again I am with the forensic personnel to discuss the scope of interdisciplinary research in the field of forensic science. My friends, forensic science itself is the interdisciplinary science. As far as interdisciplinary research is concerned, I can tell you a huge gap can be seen in the field of forensic science. And the best evidence is the forensic science laboratory manual. Once you go through the manual in the reference section, at least you will find that whatever you are doing, the tools, technique, as well as the methodology to investigate the forensic cases, basically it is 50-year-old, 70-year-old. At least this methodology, these tool techniques as older than your birthday. You know that if these techniques are 30-year-old, 40-year-olds, then one can say that there is a huge gap in the field of forensic science, because these are the interdisciplinary science. You young generation have to cover this gap. I will not going to detail in this my 1 hour lecture that the different issues regarding the forensic science in India. We cannot compare with the Federal Bureau of Investigation in the USA, because a lot of the research funding provided by Federal Bureau of Investigation, and we cannot compare with the Directorate of Forensic Science in India. My topic will not cover these all issues with the forensic sciences, but at least I am going to discuss the application as well as the interference of UV radiation in the field of forensic investigation. Basically, I will highlight the dark part of the sun because the solar radiation is, you know, that the ultimate source of energy. But today I am going to discuss how this environmental radiation having the ultraviolet UV, which is sufficient, basically the UVA, which causing the damage. And in the case of forensic, how these UV radiation are interfering with the basic results. And the so many results you have, you can come to the conclusion that there is a no any final conclusion or the inconclusive results. Starting with the basic of UV radiations, on the basis of wavelength, this UV is broadly divided into the three main heading. UVA is ranges from 320 to 400 nanometer, UVB 280 to 320 nanometer, and the lower wavelength is basically the UVC. UVA is the less energetic in comparison to UVB and UVC, but having the great penetration power. It can across your all dermis level, it can reach up to the blood capillary level. You know that it is the less energetic lower wavelength, and these wavelengths are extensively used in the field of forensic sciences extensively, because all the lower, all the body fluid, basically they are giving the special fluorescence under this lower wavelength of UV radiation. And unfortunately we are processing the same sample till the DNA fingerprinting and the STR typing. My question is that is this technique is basically the non-invasive technique. If you talk about the UVB, it is comparatively no doubt the more energetic than in comparison to UVA, having the shorter wavelength, but it is directly absorbed by the DNA as well as the chromophore of skins. UVC, we are very fortunate that the ozone layer is totally absorbed this UVC radiation. Unless and until there is a depletion in the ozone layer, it cannot come to the earth's surface. Basically in the region of Antarctic region, or due to the global warming, the few region on the earth there is a depletion in the ozone. That's why we can expect the UVC almost, but the environmental uh, radiation cannot have the UVC. UVA and UVB are more dominating. In UVA and UVB, basically UVA are dominating 90 to 95% is UVA radiation, 5 to 10% is only UVB radiation. Our earlier mindset is that only UVB is harmful for us. That's why all the safety measure regarding the UV is we have the UVB filters only, but we cannot ignore UVA simultaneously. Yes, for the forensic application, my prime focus is the examination of body fluid, because I have said that almost all the body fluid, even the seminal fluid, vaginal fluid, even the saliva, they all give the special peculiar fluorescence, and we cannot define that fluorescence unless and until you able to see it under the lower wavelength. And these all sample have been further processed for the final confirmatory test. And you know that because this sample, because this research study shows that this UV radiation is sufficient to cause the DNA damage, sufficient to cause the mutate at the gene level, we can find this mutation at the protein level, but we are processing the same sample for the final the DNA fingerprinting. There is a lot of the interference with this UV radiation, and this technique is not only in India, throughout the world people are using for the preliminary examination. Apart from that, the confirmatory examination of the drug compounds in which we are using the UV radiation, authentication of painting as well as the fine art, authenticating of the signature, analyzing the question document, currencies in which we are using the UV radiation, illuminating the fingerprints at the crime scenes, the trace evidences, even the stains as the residual stains, we are using the UV. But my main focus is the body fluid examination, because our research, I am going to discuss how this our research have revealed this successfully, that this UV, even the lower wavelength, is sufficient to cause the DNA damage. At the molecular level we able to trace it. Dear students, if you have any questions, you can stop me in between also. I will be happy to respond at the end also, because I am going to share some, because I have the background of forensic science. I did my master in forensic science from the NICFS Delhi, and I am currently the research scholar in USA. There is a huge gap, a lot of things I have to discuss in that field. If you have a question, you can stop me in between also. And because I am going to share my research work also. I am not sure how many the target audience will able to get me exactly, but even you have any question, you can ask me. There will be a response from my side. Obviously, even after this session also, because we cannot able to discuss this all thing within this the limited time period. If you talk about the source of UV radiation, the environmental source, the sunlight having the enriched UVA as well as the UVB radiations. If you talk about the UV source which we are using in the field of forensic sciences, in the case of forensic sciences, that is the portable device of the UV lamp, and we are using the same lamp, same source in my in vitro as well as the in vivo research, just to establish the how much this UV radiation is hazardous for the health impact, how much they are causing different skin diseases as well as including the skin cancer. This UV radiation is not only limited to up to that level also, because this can cause the premature aging of skin, even the sunburn, wrinkles, and the leathery skin, liver spot you can find with the help of these radiations. As well as there is a serious eye problem in the case of this UVB radiation, because UVB is more dangerous to the eye. In our India and the tropical region, so many cases of the cataract, you have heard that. These are the main cause, the main culprit is UV radiation, UVB basically. These are harmful for that. Because you know that the UV is the known carcinogenic agent, and this discovery regarding the mutation as well as the same site mutation, DNA, pyrimidine cyclobutane formation, these have been discussed a long ago, before your birth. You can say that if you are the young students, the college students, the UV is carcinogenic, and they have the carcinogenic property. You can find a lot of research article based on that. But in the forensic cases, still the all body fluids have to go through the primary screening with the help of UV only. These are some hazardous effects of the UV radiation, such as the phototoxic dermatitis, phototoxic reaction, erythema, edema, photoallergic dermatitis. As far as the skin cancer is concerned, known as the SSC, BCC, squamous cell carcinoma, basal cell carcinoma, as well as the melanoma. These are the three major types of the skin cancer, which is caused by the UV radiation. If you talk about the melanoma, although it's very rare, but the percentage recovery is very, very less in comparison to squamous cell carcinoma. Squamous cell carcinoma is very frequent, can be induced with the help of UV radiation, because in the research, so many research articles you can find that where the UV radiation is given as a positive control just to induce the tumor in the mice. The squamous cell carcinoma is very much frequent, but the percentage recovery is high. The 3 million non-melanoma and approx 1.5 lakh melanoma skin cancer, these are globally occur. These are the report of WHO reports. And the culprit is the sun exposure having the sufficient UV radiation. If you talk about the protection, how you can protect yourself from the environmental UV radiation and what are the impact of that, if you find these sunscreens, because in India also the research shows that although we Indian have the brown skin, the melanin content of our skin is comparatively enriched in comparison to the people from the US as well as the Canada and the other developed country, because they have the few melanin in that skins. We Indians having the brown skins, and this beauty of this brown skin is that they have the capacity to shield you from the hazardous effect of UV radiation, because the melanocytes having the melanin is the black color pigment. Once the UV radiation is interact with your upper keratinocytes, this melanin will come and cover your whole the skin and protect yourself from the hazardous effect of this ultraviolet radiation. And we Indians have, but the research shows that still we Indian require the sunscreen lotion with the SPF 30. These sunscreens are first developed for the soldier involved in World War II, and our mindset is that only UVB is harmful for us. That's why all the sunscreens, all the safety measure from the UV, that is level as the sunburn causing radiation, and they're having the UVB filter only. But the recent study shows that UVA is equally harmful for us. We cannot ignore a UVA. The SPF may define as is the minimal erythritol dose of UV for the sunscreen protected skin divided by unprotected skin. These are the ratio by which we can find out what are the efficacy of the sunscreen lotion you are using just for the protection from the ultraviolet radiation. And this sunscreen lotion, even in India, the all your fairness lotions and screen, you can find the 15%, at least the SPF 50 will be integrated with that, because you need the UV filter, and even you people are also using somewhat some content of the UV filter in your fairness cream also. The SPF, I mean the higher SPF means the higher protection from the ultraviolet radiation. It doesn't tell you anything protection from the UVA, but the recent study shows that UVA is equally compatible to cause the skin aging, other skin diseases, including the skin cancer. That's why recently FDA had proposed a star rating for the UVA protection, and now you can find out your sunscreen apart from the SPF as, and this a star rating. Low star is the one, and as well as the highest 4 star you can find apart from the SPF, because SPF doesn't tell you anything regarding uh, the protection from UVA. SPF is giving you some protection from UVB only. These are the skin lesion and the cancer lesion. There is a very distinct feature. You can differentiate this lesion from the mole, normal mole, as well as the skin cancer mole, because the normal mole is the very best example of the symmetry. You can divide that mole into the two equal part by the any plane, that is the normal mole. But in the case of skin cancer, this cancerous mole, you cannot divide this into the two equal part, because these are the very much asymmetry. These are the basic differences, because the people are confusing this mole or basically is it the cancerous or not, because the melanoma is very dangerous. The recovery percentage of the melanoma is very less. Here the question is how long you are safe in sunlight. I mean, after how many minutes you require basically a sun protection factor, or some kinds of the your skin will start burning. What are the time period after which your skin will start burning? The question is that these are the three dependent factors. First is what are the skin type, what are the UV index, and the sunscreen SPF of your sunscreen if you are using. Starting with the sunscreen skin type, based on the melanin content of your skin, because I have told earlier that this melanin content is vary from skin to skin, because the based on the melanin content, these skin have been divided into the 6 category. Category 1 is for the albino. A skin of albino having the very least uh, eumelanin, because there is a few melanin present in that. The skin of this albino is easily burned in ultraviolet radiation, no tanning occur. And I can tell you one thing, that tanning is the good thing. Once you expose yourself with the ultraviolet radiation, and your face and your exposed body part will become the black, this tanning is the good thing. And these are the mechanism by which the melanin will able to give you a particular color, because this tanning is not the permanent. After 1 and 2 hours it will automatically goes disappear. These are the protective umbrella you have already. That's why don't be scared if your skin is become black once you expose you UV radiation, because this mechanism is lacking in the case of albino. We don't have the true melanin. That's why always the sun burning will occur, never tanning will occur. But in the case of skin type 6, never burning, always tanning will occur, because they have the rich melanin. This thick skin is the very much protective from the ultraviolet radiation. If you talk about the Indian scenario, we Indians are categorized under the type 3 skin, which is 200 minutes divided by UV index is safe. And the UV index is the I mean the amount of ultraviolet radiation coming in your locality. These are the numerical value which ranges from 0 to 10. Higher UV index means the higher exposure risk. Minimal exposure risk having the low exposure risk. And you can find the UV index in the India also, because this is forecasted a day before in the developed country. But in the Indian context also, these indexes of the UV as well as the intensity of UV is increasing very much. There is one example by which you can find that how much you are safe in sunlight and after how many minutes your skin will start burning, or after how many minutes you need a sunscreen lotion or any other type of protective measure, because the sunscreen is not only the protective measure. Basically, if you ask me to try your best to avoid that the peak hour, because we cannot control our daily activity in the sunlight. uh, People are using the sun umbrella, people are using the sun cloth, sunglasses, and different types of the UV filter just to protect yourself. And what will be the UV index a day before that is forecasted in the developed country? Suppose your skin type is 3 and you are living in the area of the UV index 10, then your skin will start burning just after 20 minutes, because the skin type second, skin type third, go to the previous slide, you will find that sorry, you will find that if your skin type is 3, then you are 200 minutes divided by UV index is safe. And you are living in the area of UV index 10, then your skin will just start burning after 20 minutes. And if you are using the sunscreen lotion with SPF 30, then your skin have the 30 time more protection than the naked skin, and now you are 600 minutes safe. This is a part of research work. I am going to correlate with how they able to induce the DNA damage in the keratinocytes. This keratinocytes is the, I mean, superficial layer, I mean, the outermost covering of our the skin, which is protect from the all environmental contamination or environmental toxicities. These are the Hackard cell lines. These are the human Hackard cell line. I had used as a model of the testing for the in vitro model as the Hackard cell lines, and with the exposure of UV radiation, I able to prove that how they are harming our skin. The result shows that these are the base of DNA 2-deoxyguanosine. This 2-deoxyguanosine is the, I mean, guanine base of DNA, which is degraded by UVA, UVB, and sunlight. These all three components which is available in the environment, and the same intensity I have exposed in the laboratory, and we able to find that not only sunlight, even the individual UVA and UVB have the capacity to damage the, I mean, base of guanine. Further, this, I mean, damage of this guanine base have been confirmed with the specific quencher sodium azide and DEBCO. I have used as the specific quencher, which confirm the role of UV radiation in the damage of these bases. The next validatory experiment we have done with the comet assay, and this comet assay, the result is interpreted in the form of tail DNA as well as the olive tail movement. These are the parameter by which we able to confirm that yes, these are the solar UV radiations which is sufficient to cause the damage of the DNA. Further confirmatory test, I have used these cell lines for the this cyclobutane pyrimidine dimer formation, this CPD. Basically, what happened when the two thymine, I mean the two pyrimidine, if it is come together in the adjacent one base of the, I mean the one strand of the DNA, then in the presence of ultraviolet radiation, these two thymine will get dimerize itself and break their bond to the complementary adenine on the another complementary strand of the DNA. That's why this DNA is get mutated, and this dimer can be find almost all the site of the skin cancer. That's why this pyrimidine formation is called the UV signature mutation. And in the case of if this the cancer, skin cancer is the culprit is UV radiation, then there is a high chance that you can find the same site mutations. Further, we have found another experiment based on which we able to confirm that this UV radiation is not only the genotoxic, as well as they have the capacity to induce the reduce the survival ratio as well as the apoptotic parameter. They have the severe impact on the mitochondria and as well as lysosome also. Further, for the confirmatory experiment, we able to expand our research work till the gene and protein level with the help of real-time PCR. We able to find out there is a activation of these apoptotic marker genes as well as the further confirmatory I have reached to the Western blot with the help of a protein. We able to find that this is not only at the gene level, even at the protein level, this damage is caused by the UV radiation. The caspase 3 cleave as well as the pro-caspase, these are the, you can say that the these if these are activated, these are the marker of apoptosis and which is induced by the UV radiation. And this study, our study, which is already published in toxicology later, confirmed that the UV is not only the genotoxic, they having the property of the cytotoxic also itself. That's why the genotoxic and the cytotoxic property of this UV radiation have been confirmed by my study, and which is already published in the Journal of Toxicology later. Another study, the extension of this study, our group in the Yale University have this study, and these are the very interesting study which confirm that the UV induces the delays damage of the DNA, I mean the cyclobutane pyrimidine formation in the mouse melanocyte. These are the very much interesting studies, because till date we have the information that once the UV radiation is incident in your skin, there is a instant damage of the DNA. But this study shows that even once you end the radiation, still the damage is continue in the case of mouse melanocytes. And this study is very interesting, and which have been already published in the very good journal in the Science, the name as Science, with the help of Brass as well as their team. Here the figure A shows that the albino melanocytes is these are the melanocytes having the not the true melanin, they have the few melanin. And in the case of few melanin, we didn't find any CPD formation higher than 0 hours. But in the case of black melanocytes, this damage is continued till 4 hours. You can see here, this damage is higher than 0 hours. It means that once you off the radiation, once you off the in the exposure of UVA, the damage is continue. This is the very first time reported this study, the delayed CPD concept. The further when you expand the timeline, because you cannot show that the exact what is it is the 3 hours or 4 hours. Once you expand this timeline, you able to find that these, I mean, we are getting the delayed damage higher than 0 hours till the 4 hours, and the maximum at the 3 hours. Further, this study have confirmed with the, we have able to silence the gene responsible for the DNA damage, because we have the repair system, repair mechanism. Once your skin, once your DNA is get damaged by the radiations or any other active agents, there is a auto system we able to repair itself also. Once we knock out this gene which is responsible for the repair of the DNA, excision repair, you will able to find that this damage is continue till 2 hours. Even if we are talking about these albino mice, because in albino mice this repair mechanism is very much active, and just after 0 hours, just after the exposure, the repair is very much active. That's why we didn't find anything higher than 0 hours. But in the case of black mice, we able to find that this same albino mice, once we knock down the gene which is responsible for the damage of DNA, we able to find that this damage is continue till 2 hours. Further, this study have been extended to the human skin. The epi skin of the human is collected from the hospitals, and which is irradiated with UV radiation, and we find that although there is a huge variation in the experiment, no doubt, but we able to find that still there is a chance of the delayed CPD formation. I mean, once you switch off the light of the source of the UV radiation, the damage is continue till 2 hours. The non-producer of this CPD, I mean the non-producer means the guy which is not able to produce this CPD till the 2 hours and 3 hours, and the maximum at the anything more than the 0 hours. Again, when you knocked out the gene responsible for this DNA repair mechanism, we able to find that this damage is continuing. And you can see here the maximum damage is at 2 hours, because the repair mechanism is very much active in the case of non-producer as well as in the case of albino mice. That's why we're not able to find out this CPD formation again at the 0 hours and 2 hours. These are the transgenic mice which can reveal that very clearly that this CPD formation is higher at the 2 hours in comparison to 0 hours, which can be reveals that this CPD formation in the case of melanin, in the case of I mean the black melanocyte, these are the continue till 2 hours, 2 to 4 hours. Further, the exactly this mass spectrometry reveals that the intensity of the dark CPD and what are the different categories, different classification of dark CPD, this thymine dimerization, as well as the thymine cytosine, cytosine thymine. There are the three categories. These all are comes under the cyclobutane pyrimidine dimer, and we able to find that in the case of albino mice nothing is more than 0 hours. It means that at the 0 hours the repair mechanism is very much active. That's why we didn't find anything higher than 0 hours. But in the case of black melanocytes, we able to find that at the 2 hours that is more than 0 hours, and this thymine dimerization is very much dominating in comparison to other type of, these are the thymine cytosine thymine. These all three are comes under the cyclobutane pyrimidine dimer, and which is exclusively formed in the case of light only, because the chemical agent cannot able to produce that. That's why these are the confirmatory tests that we can assure that the culprit is UV radiation only. Although these radiations having the other positive impact also, but we are highlighting the dark side of the sunlight, and which is sufficient in with the case of UVA. And UVA is the lower wavelength, and that lower wavelength is ambient in our environment throughout the world. Throughout the world, even in this COVID pandemic, this intensity of UV is increasing throughout the world, even in New York City, as well as the intensity in India and the Asian country also we able to find this and this same lower wavelength we are using in the case in the field of forensic science, just for the examination, at least for the examination of all body fluid, and we are processing the same sample for the DNA, fingerprinting, STR typing, and the other confirmatory tests. Next experiment, they able to find out, the Premi et al, they able to find out what are the exact mechanism by which this damage is continue till 2 to 4 hours, and we able to find that there are the three culprit, three dependent factor, and the major one, first one is the melanocytes, I mean the synthesis of melanin, and which can be inhibited by the kojic acid. This kojic acid is something, the chemical which can inhibit this melanin synthesis, I mean the melanocyte synthesis, and this synthesis can be stopped with this kojic acid. We able to find that once we inhibit with the help of kojic acid, there is nothing higher than 0 hours. I don't know you student will able to get this research clearly or not. If you have a doubt, please note down, and you can stop me in between also, because I thought that my target audience is most of the student only, and I don't think the student at the BSc and MSc level will able to get it exactly what are this research. But still you have a query regarding the basic question, you can ask me, I can explain it in detail. Is it okay?
Sir, there are two questions in chat.
Yes, yes.
First
I
one
not.
is
Yes, yes.
first one is UV is helpful and after afternoon UV is not. What is your views on it?
Just repeat the question.
UV is helpful and afternoon UV is not. What is your views on it?
Yeah, UV is helpful because the sun bath are there, sun tanning are there. These are the protective mechanism. You know that only with the UVA radiation lower wavelength. But if you are exposing with UVB and the higher wavelength, it are very much dangerous. That's why afternoon is not, because the higher intensity, the peak intensity can be recorded at the 11 to 2 p.m. I mean 11 a.m. to 2 p.m. These are the peak intensity, and the most of the daily activity will happen these peak hours. That's why you have to avoid this. Yes. What are the next question?
So second question is, in Africa, tan people also suffer from skin diseases. What is the exposure risk to them?
Just repeat again.
Sir, the question is, in Africa, tan
Okay.
people also suffer from skin diseases. What is the exposure risk to them?
Yeah, very much interesting. You know that these 10 people, I mean, the thick skin having the, I mean, true melanin, these are the protective mechanism. It doesn't mean that you cannot have the any skin ailment. 20% people diagnosed with the skin cancer in USA. I mean, you can say that the one in five people, I mean, one in five people must be diagnosed with the any kind of skin cancer. In Africa, they have the protective mechanism, but this study shows that our study, if you able to listen this study clearly, the people, I mean the guy having the melanin, I mean the rich skin, their skin, I mean their DNA is damaged, is continued till higher than the 0 hours. In the case of albino, this the last slide, in the case of albino, it is clearly shows that they are safe here. The albino means they have a few melanin instead of eumelanin. Eumelanin is the protective melanin. The guy is safer because whatever the damage is till 0 hours only. But in the case of melanocytes having the rich pigment, this damage is continue. And if you able to correlate this experiment, this study with this guy having the, I mean, Africa and other country, even in India also, a lot of our South Indians, you know that it doesn't mean that the South Indians having the, I mean, high concentration of melanin, they doesn't have any skin diseases. It's not like that, right?
Okay, sir. Sir, there is one more question. What are some simple steps to protect oneself from skin cancer?
Oh, I cannot tell you in one line the what are the simple steps, but you have to be careful. What are the UV index of the day before, and you should accordingly prepare yourself because in India the forecast is not exactly. In US and the other developed country, they will give you the exact, I mean, forecast of the UV intensity. What will be at the what time? Even the hourly intensity of UV radiation,
है
based
वो
on
चालू
which
था
you can protect yourself. Try to avoid the peak
गुड
hours.
मॉर्निंग गुड
Try
मॉर्निंग गुड मॉर्निंग
try to avoid the peak hours. Try to use
नहीं
the
2 दिन से तो
sunscreen
मैं घर पे था
lotion and the I
एकेडमिक
mean the best sunscreen lotion, because in the market the chemical lotions are there which having the so many allergic reactions also. That's why try your best to avoid these peak hours. Try to use the, I mean, a full sleeve cloth. Try to protect your body as much possible. Try to use the sun umbrella, sunglasses, even the sun cloth is available. If you are using sunscreens, to be very cautious regarding the choose of these sunscreens, because so many UV filters having the hazardous effect on the skin allergy as well as the phototoxic dermatitis. This can be enhanced with the help of sunscreen lotion. Try your best for that. Yes. Yeah.
Sir, there is one more question.
Yes.
What is the best sunscreen will you suggest for use?
Oh, I cannot say directly to the brand and this platform, but try your best for the go with the herbal sunscreens. And if the label is only herbal, you cannot claim that there is no any chemical ingredients in that. But try your best to go with the herbal sunscreen lotion. I have done few research. I cannot reveal the brand of this company, but they are claiming the herbal, but having the chemical ingredient inside that, right? Go with the herbal extract. The few company in India, they are providing the herbal sunscreen lotion comparatively better than the other organic and inorganic UV filter they are using.
So there is one more question.
Yes.
If ozone layer gets thick again,
One more question from my side. These questions are by the student or the faculty member? All students,
yes,
right?
yes, sir, from the students.
Oh, that's good. I am happy that the student have listened me at least, because in the very beginning I thought that the target audience is the largely a student. Dr. Rajiv shared with me that we have the 250 student, and I see here the 100 participant. Mostly a student will be there. They will not able to get it, but I am happy that you are getting something from me. Yes, please go ahead.
Yes, sir. The question is, if ozone layer gets thick again, will UV affect us in high temperature?
The temperature and the UV radiation is the two different factor, because the UV radiation increase the thermal effect also, because there is if there is no any sunlight, there is no any this intensity of UV is also less. If the ozone layer is, your question is if the ozone layer is able to heal properly, right? Hello.
Yes, sir. Thank you so
Yeah,
much.
if the ozone layer is a proper healing, then there will be no any UVC only. But I have tell in very beginning that the UVA and UVB will able to cross the ozone layer, because the absorbance of ozone layer and the UVC is equal. That's why ozone layer able to absorb all the UVC radiation only, and UVA and UVB is equally harmful for us. We cannot ignore. Very unfortunate is that the UVA is equally important, and they able to, I mean, cause the DNA. Although you guys have not asked me the question, because the observance of DNA is 260, right? And the protein is 280. This 280 to 260. But if you talk about the wavelength of the UVA radiation, which is starting from 320 to 400 nanometer, right? But if this observance is not come in that region, because UVA is 320 to 400 nanometer, these are the lower wavelength, but still they able to cause the DNA damage, severe damage. Although the observance range is not included in that. But what are the exact reason? Scientists are still going in that detail, but not able to find that. But we know that I have done the experiment, many experiment, that which is the clear proof that the UVA is sufficient to damage the skin. But what are the mechanism by which we able to do? We are still waiting for that. Okay, can I proceed to the next slide? And you have the more questions?
Yes, sir. We have three more questions actually.
Okay. Yes, I am happy to answer the first question. Go ahead.
Okay, sir. The question is, as UV damages DNA, can lasers be employed in extraction of DNA with certain adjustments in future as advanced technology, or lasers too can damage DNA?
Obviously laser, because this all research is this laser cannot be used for the extraction of DNA. Because this DNA, they if they able to fragment in, then what are the purpose of extraction? You have to try your best, whatever the things you are extracting should be intact in all respect. Then I don't think that a laser will able to help you in the extraction. We have a very smart thing in US, even in 1 minute, even in 2 minutes, we able to extract the DNA, we able to extract the nucleic acid. I will show you the few instrument by which we can go through very quickly with the help of that. But this laser will not help you in the extraction, I don't think so. But I cannot tell you 100% sure that what will be the future outcome of that. But right now, for me, the lower wavelength and the laser is something damaging for that. So you have to try your best to avoid the sunlight, even during your extraction procedure. I am trying my best to least exposure with the even sunlights, as well as the lights, tube lights of the I mean the lab also. Yes, next question.
So next question is, why some mutation in very important genes is related to some cancer and not the other? You might say this depends on the gene function on the specific cell. So why mutations in DNA repair genes are associated with specific cancer and not the other?
Your question is because this each and every gene have the specific function. You know that we have the excision repair system, and our study shows that once we stop that the repair system, the DNA damage is continue, and there is a specific gene for that also. All the mutation, all the genes have doesn't have the diverse function of that, the specific function. That's why the gene have categorized based on that, and we able to find so many genes which is responsible for a DNA damage. In the case of DNA damage, we able have having the higher expression of that gene. Yes.
Yes, sir. So next question is, according to the skin type chart, We should not spend approx 1 hour under sun, but we have seen people spending appreciable time under the peak hours of sunlight, even though they don't suffer from any sort of problems.
Okay. The thing is that these all are the not the instant problem. Whenever this skin aging, you know that this skin aging is the impact of this ultraviolet radiation. uh, 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 I mean, the what you mean by the people should you able to find out the DNA damage or not, because the guy is exposed with the 1 hour and 2 hour in the sunlight. The thing is that the leathery skin, liver spot, elastosis, different segments of the skin aging and the skin diseases, and which is cannot be seen by the acute exposure. These are the chronic exposure. Daily, if you are exposed with the 20 minutes, 30 minutes extra, then your the capacity, then your the I mean concentration of the U melanin you have, you must have will be the problem in the future. They are not the acute effect. Once you go with the UV exposure and you will find something mishappening with you, it's not like that. Body have itself the protective measure, body have the repair mechanism also, but the skin aging for sure, no doubt, these are the irreversible. We cannot able to stop that. The leathery skin, elastosis, keratosis, and the other dermatitis, this can be happen in future, and these are the accumulative effect also. These are exponentially grow. That's why try your best to avoid this thing. People are not serious about that thing, but this will come in future, because in old age, in the once you go above 40, once you go above 50, you know that these all person with the leathery skins, as well as the skins, this liver spot on that skin, elastosis, these are not uniform, and this doesn't happen the all population together. That's why try your best to avoid it. You have to delete this thing from mind, that this UV radiation cannot do this up to that level. These are the accumulative effect. Yes. Yes, please.
Thank you, sir, for answering all the questions. I think,
Okay.
yes, sir.
Okay. Now I'm going to reveal that how this DNA damage is continue even your skin is more protective, because the black skin having the more melanin, I mean the more true melanin in comparison to few melanin. And our mechanism shows that the three factor, melanogenesis, as well as the superoxide, and the third factor is the nitric oxide. These all three independent factor, we able to inhibit this factor, then we find that something higher than 0 hours. And further we confirm with the help of this triplet state quencher. And in the triplet state quencher, these are the CPM count per minute. We able to find that DBAS is a probe which is accepting the triplet energy only, and we able to find that these are the triplet state quencher by which we able to find something higher than 0 hours, which is happen in the case of, I mean, the true melanin, in the case of mice having the true melanin. And these are the proposed mechanism photochemical pathway which shows that the UV radiation can able to cause the inos, which is the further goes convert into the nitric oxide and the superoxide. This nitric oxide and superoxide combined together come to the peroxynitrite, and this peroxynitrite target your melanin. Even you have the true melanin, although, because your question was the why the people from the South Africa having the rich melanin is still having the skin problem. These are the answer of that question. This peroxynitrite target the melanin only, not the few melanin. They targeting the true melanin, right? And this fragment of melanin goes and diffuse inside the nucleus, and with the help of peroxynitrate as well as the degraded melanin, they able to excite the energy level of the triplet state energy level. And this triplet state is very short time period but very highly energetic. This energy can be captured by the DBAS probe, but before they get captured with that probe, they able to damage the DNA. These are the one strand of DNA, and these are the pyrimidine. Suppose these are the thymine and thymine, or either thymine and cytosine. They break their bond to the complementary adenine and get bind together, and these are called the CPD, cyclobutane pyrimidine dimer formation. And this dimer, this same site mutation is seen in the case of a skin cancer. That's why we are called it the UV signature mutation. UV signature mutation, suppose one thymine and one cytosines, they are the very much frequent. That's why it's called the UV signature mutation. These are the proposed mechanism. These are the paper which I able to publish, which confirm that the UV radiation having capacity to damage your DNA. I have published in the toxicology letter. The another work have been published in the same journal. And because most of you are the student, I am going to have the lab tour. I mean, the what are the equipment and we have in our lab, how we are doing the research work. We can ask the question based on that also. These are the real-time PCR. With the real-time PCR, what we are doing? Anything from a student side? Any student? What do you mean by real-time PCR? What do you mean by PCR?
The PCR is polygonal.
Yes, 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 please. PCR is?
Sir, PCR is polyacyl combined.
What is PCR? Any one of you?
PCR
Basically,
is poly.
polymer
Poly-
chain reaction. Polymer
Yes,
chain
sir.
reaction. Basically, what happened in the case of DNA? The quantity of DNA is very very less. That's why for the further study, first you have to amplify this DNA. You're not able to see the DNA with naked eye, right? You're not able to see your one hair.
Yes.
Suppose your one hair is fall on your notebook or your I mean your phone even or your MacBook. You're not able to find, but the same hair, same piece of the hair, suppose 1000 same piece, same hair, if it is come together, you able to find it very clearly that this PCR is doing the same thing, because they are amplifying, they are just copying the same thing again and again, again and again. This instrument is called PCR, polymer chain reaction. Basically these are the thermocycler based on the different different, I mean the temperature differences. This one DNA divide comes to the two DNA, 2 to 4, 4 to 8, and 8 to 16. Further, these are the exponential increase 2 to the power n number of DNA. You able to get it? What are the purpose of that? Because we the quantity of DNA is very very less, we not able to visualize in the gel, we not able to detect without the amplification. That's why we you need something to amplify it first, that is called the PCR, right? Here we are using one fluorescent dye, with the help of that we able to quantify it. And these are the, that's why I call it, these are the real time PCR. The real time PCR means you can stop the how many requirement you have, what are the cycle number in the between the process we able to find. But the conventional PCR, you don't have any idea your sample is I mean amplifying or not. That's why it's called the real time. At the meantime you can stop also, you can find that what are the amplification, how many cycles occur, everything is on time. That's why I call the it's the real time. The next we have the DNA microarray. With the help of real time PCR, we able to amplify the gene, we able to find out the gene expression, I mean the 10, 20, 50 gene, 100 gene. But in the case of DNA microarray, you can find the expression of 1000 genes together. These are called the DNA microarray scanner. There is a chip in which you can study the thousands of his gene in one go. But in the PCR, you cannot do the thousands of genes. These are the basic differences in that. The next is the MI-seq. These are the, I mean, sequencing. MI-seq, the sequencing technique. You have to align. You have to find out the sequence of DNA and which DNA, which segment is the damage. What are the, I mean, how many bases is damaged. Based on that, we again we are using the fluorescent dye based on which we able to find out the what are the sequence of the DNA. And the next we have the next generation sequencing. These are the high output. You the simple difference between the MI-seq and next generation sequencing is that you can say that the this efficiency of this next generation sequencing is 10 times higher than that. For the whole genome sequencing, if you have the large experiment, this MiSeq will not work, but this next generation sequencing will work out that. The meaning of sequencing is nothing. What are the sequence of your DNA, sequence of the nucleic acid in that in one go, because you guys are not able to get exactly the what are the function of PCR. I read it first. Again, if you have a question why we are doing this, then I will ready to explain you, and there will be a response from my side. You can note down my email ID as well as my number. There will be a response from my side. These are the single cell genome sequencing. I mean the single cell, I mean you can here use a template of the single cell for your miRNA, I mean macro RNA sequencing, your mRNA sequencing, your DNA sequencing with the one cell only. That's why it's called the single cell. The one cell having the concentration of DNA is very less. That's why in able to compare with that. The macro injector, these are basically we can edit the genome. With the help of that we can insert something of the desire inside the zygote, and the output will be accordingly of that. The genome editing will happen with the help of this micro injector. These are the gene pulsar. Basically the transfection, I don't think the student will able to know these things. Suppose you have to insert something of your interest, this called the transfections, and with the help of these you can transfect almost all type of cells, right? And these are my personal opinion that the what are the research and development barrier in the forensic science. Although these are my personal opinion, the lack of exclusive funding agency. If your project is based on only forensic theme, then who will going to fund you? These are the biggest problem. That's why the gap in these interdisciplinary research in the field of forensic science. The second thing is that the lack of forensic research institute. We have the so many institutes, we have the so many academic institutes, no doubt, but we don't have the any exclusive institutes which are focused on research and development in the field of forensic science only. The third important parameter is the police and the forensic department. They are not on the same page. There is a differences in that. There is a thought process is differ, and we not able to establish a proper coordination between that. These are the reason of the huge gap in that. The number of pending cases is one of the most important reason, because if the research scholar will get join a good forensic science laboratory, still they're not able to do their research work. The thing is that they have the severe workload. They have to focus on the pending cases only, not their research work. For query, you can Google me. My name is Saroj Kumar Amar. You will find these all environmental toxicity related questions, as well as the what are my research article. Based on that, you can directly contact me with my Gmail ID also, and my phone number also. Many thanks. Thank you. If you have any question, please.
Yes, sir, there is some questions.
Yes.
First one is, what is meant by 0 hours?
0 hours, very good. 0 hours means the exposure when you end is called 0 hours, because the study is called the till date we have the information that the damage is instant. Once you expose something with DNA, something with UV radiation, the damage is instant. And once you stop that exposure is called the 0 hours. And the 2 hours means after end of exposure 2 hours. 4 hours means the after end of exposure till 4 hours, this damage is continue. Yes, please. Next.
Yes, sir. Next question is, how many UV signature mutations are there?
I have show you in the slide that basically the three types is a major common: thymine, and the UV signature. If you talk about the UV signature, then they are the thymine and cytosine, right? But the thymine, cytosine, thymine, and thymine, cytosine, these all four categories are called the pyrimidine dimer, right? These all are the pyrimidine. Whatever the dimer forms between the pyrimidine is called the cyclobutane pyrimidine dimer, but the UV signature mutation is the cytosine and thymine. Yes. Next question.
Thank you so much, sir, for such an interactive session. I now
Thank you.
Yes, sir. I now request Ms. Kajol Bhati to facilitate our guest.
Thank you so much, sir, for such an interactive session. You have explained UV-induced DNA damage so nicely, and you explained all the questions with patience. And I thoroughly enjoyed the session, the benefits of tanning and everything. People don't understand that, but yeah. So thank you so much, sir. Please accept the token of
I will
gratitude
take
from
care
our
of that.
side.
It's a very good. Thank you for the certificate. Ask Miss Binni Sharma that please mail the certificate to me.
Yes, sir. We'll definitely mail you the certificate.
Okay.
Thank you so much, sir.
Thank you.
Thank you so much, sir. Thank you so much, ma'am.
Thank you. Thanks a lot.
The next speaker for today is Dr. Desiree David, lecturer, Department of Law, Nelson Mandela University, attorney at High Court, South Africa, on the relationship between science and law and the interest of science or interest of justice. Ms. Desiree David is attorney of the High Court of South Africa, lecturer in Department of Criminal and Procedural Law, Nelson Mandela University, South Africa, and also director at South Africa Medical Legal Association, medical malpractice mediator, street law trainer and facilitator. Her area of expertise is law of evidence, health law, legal practice. Ma'am, I request you to kindly take over the session.
Good morning, everyone. It's very early in the morning here in South Africa. Thank you very much for giving me this opportunity to address you all at this wonderful week of very informative sessions. I must admit, coming from a legal background, I've been joining some of the sessions, and it's very wonderful to hear what is happening in your field specifically. So thank you very much for giving me this opportunity to share my thoughts on interdisciplinary research and interdisciplinary relationships, specifically in the context of the justice system. So, sorry, just excuse me. I have dogs with me and they are making a noise for some reason or the other. I will try to shut them up. As you heard, my work has been very much in the legal field. I come from a litigation experience, a litigation background. So I've spent a lot of time in the courts. And what we have seen in the last 20 years or so is the fact that in court cases, both in criminal as well as civil cases, there's been an increasing reliance on the use of forensic evidence as part of the court proceedings. So in other words, we are relying more often on the use of forensic evidence as part of the evidence that is presented in court via expert witnesses. If you think about traditionally how we received evidence in court, it was via witnesses who would give testimony. So like eyewitnesses or people who knew something about the issue. But what the research has found is that eyewitness testimony is increasingly unreliable because there's so many different factors that go into an eyewitness's testimony. You know, there may be implicit bias in the testimony of an eyewitness, things like education, things like financial position, things like your position in society, your social standing, all those kind of things could have an impact on eyewitness testimony. And so eyewitness testimony was seen as being unreliable. When you have evidence, forensic evidence, when you are relying on the science from an unbiased, totally impartial witness who's analyzing the scientific evidence and making and reporting those findings to the court, it is seen as being more reliable. So even in some cases where, for example, evidence may be treated as circumstantial, it is still afforded a lot of weight by a court when it's weighed together with other evidence that is presented. As you know, my expertise is in evidence. Our legal system is very similar to what you have in India. I think because of our colonial past, we've had the same or a similar kind of colonial history as what you've had. Our law of evidence, although our legal system in South Africa is based on Roman Dutch law, our law of evidence is based on the English law of evidence system. So there's a lot of similarities between our system and your system. I just saw a question that popped up. Why is eyewitness testimony given any value if it is unreliable? So I'm going to address that right now just to set the scene a little bit, because I understand you guys are not legal people. So when a judge has to make a decision on a question before the court, the judge listens to all the evidence that is presented, and then he will evaluate and weigh up the evidence. So when we talk about evaluating the evidence, say, for example, you have an eyewitness, right, who comes to court, he's asked questions, and he talks about what he's seen or what he thinks he has seen or observed. Then that witness will be cross-examined. Cross-examination, the whole point of cross-examination is to discredit the witness, to show him as unreliable and so on. Because if you're discrediting the value of his testimony, if you're discrediting his reliability, then the judge will place very little weight on that evidence. So when he is evaluating all the evidence, because the witness has shown himself as being unreliable or not credible or not believable or not a good witness, then the judge places very little weight on his testimony. So it's almost like that testimony, because of its unreliability and so on, will have very little value. You know, you may have an eyewitness who is so convinced that he saw Mr. X at the scene of the crime stabbing the victim. Okay, that's his eyewitness testimony. He's saying under oath, I saw Mr. X. Mr. X had a knife in his hand and he was stabbing the person. And Mr. X says, his defense is, but I was not there. I was out of the jurisdiction of the court. So you have other testimony or other evidence that comes in. So let's say, for example, Mr. X says that he was in another part of the country. And you have his boarding passes to show that he boarded a flight and he was somewhere in the air at that time. Or if you have somebody else that places him at a meeting in another area where he couldn't possibly have been doing the stabbing. So the eyewitness is testifying that I saw him there, and that's fine. But the other evidence that is being presented, the evidence of the fact that he was flying somewhere or he was in a meeting somewhere or whatever, then discredits or shows the eyewitness testimony to be unreliable, because you have some other evidence that's stronger than the eyewitness's evidence that says he was there. And so when the judge is evaluating all the evidence as to whether to see whether Mr. X is guilty or not, then the judge will say, look, Mr. X is saying this, but we have stronger evidence in the form of his boarding pass, in the form of somebody else who can document, let's say the meeting was recorded between Mr. X and somebody else on that date and that particular time. Then that eyewitness testimony is shown to be unreliable. So I hope that makes a little bit of sense to you. So when the evidence is weighed up together, this is what happens. And this is one of the reasons why it is forensic evidence is so important in these kind of matters. And in South Africa, we see in cases, for example, or in murder cases, if you know anything about South African society, you know that we have a very, very high murder rate in South Africa. We have one of the highest rape statistics in the world. So we have a fair amount of violent crime. And so we are finding that more and more we are relying on forensic evidence, especially in sexual offenses cases. One of the sad realities of South Africa is that we have a very high rate of child sexual abuse and child rapes. So you sometimes have a situation where the DNA evidence, for example, is the only evidence that can secure a conviction where a child has been raped. Because although a child is allowed to testify in court proceedings, so, you know, you may have a situation where a 6-year-old, for example, was raped. And that 6-year-old should be allowed or will be allowed to testify in court proceedings, but only if the court, if the judge believes that the child can understand the proceedings, that the child understands what it is to tell the truth or to tell a lie, because the oath is not administered to a child at that particular age. They have to affirm that they understand what it is to tell the truth and so on. So sometimes you may have a situation where, because of education, because of communication difficulties, because of not being not understanding the gravity of what a court case is about and so on, the child may not be competent, may not be allowed to testify. And in that kind of situation, the child and the rapist is the only two people that would have been present at the commission of the crime. So the problem is, if you don't have a child who can testify, or maybe because of trauma, the child is not in a position to testify, all you are relying on is the scientific evidence or the DNA evidence. So you want to be able to make sure that that evidence is as accurate and as good as it possibly can be, because you want to get that particular rapist off the street. So the previous speaker alluded to the fact that sometimes you have this disconnect between the police and the forensic scientists or the experts. You have the black backlogs at the forensic sciences laboratories. From the legal perspective, I can tell you that that is the case in South Africa, as it is in a lot of different jurisdictions. So what would sometimes happen is that the police, because of lack of training or because of not exercising proper care and so on, don't follow the proper protocols when it comes to collecting the evidence and so on. So there's a break in the chain of custody. And the evidence, the biological evidence, for example, reaches the forensic sciences laboratory. The scientists will then do the necessary tests and so on, and then they will have to testify in court proceedings about their findings and about the testing that were done and so on. So they would be the experts that have to testify in court proceedings. So here you see that relationship between the legal field and the forensics field. The problem is when you have this kind of disconnect between the police and the forensic scientists, is that if there was a break in the chain of custody, so if the biological sample, for example, the DNA sample, was unaccounted for a certain amount of time. So let's say, for example, the swabs were taken, and the guy, the person who's responsible in the police, threw it in the back of his car, went out and had lunch, and then showed up with the specimens at the forensic sciences lab. And you test, the scientists the expert tests, come to court to testify. So I'm the cross-examiner. I'm cross-examining the state's witness that is saying that the DNA belongs to the accused person and so on. The first thing that I'm going to ask about is whether the chain of custody remained intact. In other words, was that biological sample accounted for at each step of its journey, from the time the sample was taken from the, let's say from the victim, up until the time it was opened and tested at the laboratory. And if you can't account for its whereabouts or its security at any time during that process, then even though you as the forensic scientist has done your work to the best of your ability, following all protocols and that kind of thing, I'm going to argue that it is possible that in the 1.5 hours that the guy was having lunch, when it was left unattended in his car, that it could have been tampered with, it could have been switched, anything could have happened to it. And so what happens in that situation is that the judge may say that the evidence is unreliable because there was a break in the chain of custody. And so that evidence gets thrown out of court. And very often, like in the case of small children that I told you about, where the DNA evidence is the only evidence that could convict an alleged rapist in that particular case, you have a situation where the person walks free. So we had a case in South Africa probably about 5 or 6 years ago. It was a case of a young girl. She was about 6 years old, and she was raped allegedly by the accused person in a particular in that particular trial. And her, when the medical examination was performed on her, certain swabs were taken. And I can't remember what happened with the swabs, but there was DNA, there was semen found on her underwear. And so her underwear was labeled and sent off for testing for DNA. But what had happened was that for those of you who watch crime dramas or who've been involved in this kind of work, you know that there are sexual assault evidence collection kits. And so each item of clothing or whatever that is taken needs to be properly labeled, bagged individually, and so on, so that there's no cross-contamination. So what happened in this particular case was that the panty was removed off the child, and it was placed into an evidence collection bag, all correctly done. But what had happened is that each evidence collection kit has a number of stickers with a specific number identifying it as being part of the evidence in that particular matter, in that particular case. And for some reason or the other, nobody knows what happened there, but the sticker from another evidence collection kit landed on that evidence bag. And so the semen that was found in the panty was tested, and it came back as a match to the accused in that particular case. But what had happened was that the lawyer for the accused person questioned the different numbers. So what he said was that the number of the sticker that sealed that particular bag was different from all the other samples that were taken from that particular case. And so he said that there was some doubt as to whether the panty actually came from that particular case because it was labeled with the case number of another case. And unfortunately for the young child in that situation, the alleged rapist walked free because the DNA evidence would have been the only evidence that would have convicted him. The child was in no position to testify. And, you know, had the evidence been properly labeled, it would have been a slam dunk. He would have been convicted. Unfortunately, that was the case in the case that I told you about. When the previous speaker also talked about the fact that there is such a backlog at the forensic sciences, at the laboratory sometimes. And he talked about the fact that you may be not going to get a chance to do your research because there's all this testing that needs to be done and so on. So from the lawyer in me looks at it from the other side, because, there's the other side of the coin. When the testing is backed up, everybody in court is waiting for the results in order for the evidence to be presented and so on. In South Africa, our constitution guarantees everyone the right to a speedy trial. So that is one of the sections that is contained in our Bill of Rights, where every accused person is guaranteed the right to a speedy trial. And there's been a few cases in South Africa where accused people, especially where they are not let out on bail, and sometimes for a person who's got a rape charge over them, it's a little bit difficult for them to get bail. So they're sitting in prison while they're waiting for their case to get to court. And very often the prosecutor comes back and says, well, we're still waiting for DNA results. We're still waiting for DNA results because of a backlog at the forensic sciences laboratory. And what happened in more than a few cases is that the judge who was hearing the case said it's unacceptable that we're waiting a year and a half or whatever for DNA results. And so, what has happened in those cases is that because the accused has argued that this long wait for the DNA results is actually infringing on his constitutional right to a speedy trial, the charges have been withdrawn in that particular case, and the alleged rapist walks free until they can bring charges against him again, maybe in future. But you can see why this may not be in the interest of justice. And one of the things that I'm talking, or that I want to talk about, is how the interdisciplinary relationship between the two professions, that of the forensic sciences and the legal profession, can actually further the interests of justice, especially when we are dealing with criminal trials like the ones that I've been talking about, which are somewhat of a heinous nature. So when I was introduced, you heard about some of the things that I'm involved in, some of the things that I do. So as I've mentioned, I've come from a litigation background where I did quite a bit of criminal work, but I did civil work as well. At the moment, I am I'm lecturing at the law faculty, so I teach forensic evidence and evidence. I teach health law and so on. So I'm involved in those aspects, and that is where we see a lot of the need for interdisciplinary relationships between the two professions, especially what I concentrate on at the university is the health law or the medical profession and the legal profession. But because of my work with the South African Medical Legal Association, I'm a director of that organization. What that organization does is to try and facilitate relationships between the medical, legal, as well as between the forensic and the medical world and the legal world, specifically in the from the point of view of experts who have to testify in trials, to provide the necessary framework for an exchange of knowledge and understanding of the different professions. Because I think the scientists and the doctors and so on generally meet lawyers in court, the relationships between lawyers and scientists and doctors is not very, very friendly, because the scientists and doctors are invariably on the receiving end of cross-examination in court. And so that relationship is not always a good relationship. I also do a lot of training work for a nonprofit organization called Street Law. And one of the things that we do is to introduce doctors, for example, or expert witnesses and so on, to the court setup, the court system, so that you actually understand when you have to testify as an expert in court proceedings, what is your function in the court? Why is it that we have to ask you these 1,000 different questions before we get to what we need you to say? Because when you have to testify as in let's say in a criminal trial, as a scientist, as an expert in your particular field. So let's say your field is DNA, and you've done the testing, or you called to give an opinion on something, you're approaching the court as an expert. Now, in South Africa and, you know, in the United States, in the UK, I'm assuming in India as well, expert opinion or your the expert's witness is seen as the expert's evidence is seen as opinion evidence. And for those countries that follow the English system of evidence, opinion evidence is generally inadmissible, because it's based on nothing but your opinion. But when you are an expert in your field, then your opinion as an expert is used in court proceedings to assist the judge in areas where a judge necessarily may not have the expertise. So a judge is an expert in the legal field, but when it comes to DNA evidence, when it comes to blood stains, blood spatter, when it comes to ballistics and so on, he does not have the type of expertise that is required for him to make very important decisions on very, very specific scientific aspects of the trial. So as an expert witness, your function is to be unbiased, impartial, and your function is to assist the court in matters where the judge does not necessarily have that expertise. So the judge, he listens to you, to what you have to say and so on, even though it's your opinion. It's based on your years of study, your years of experience in that field, your publications, how your reputation in your field of expertise and so on. So when you have to testify, you will have to establish your credentials. You will have to establish your expertise. And that is the reason why us lawyers will ask you the 1,000 questions, because what we're trying to do is to establish to the court that you have the necessary expertise and that you are in a position to assist the court when it comes to these kind of matters. And especially as I spoke about earlier on, the fact that we are relying more and more often on forensic evidence, on the scientific evidence, it becomes very, very important where, you know, you may have a lot of, you may have eyewitness testimony, for example, that I said may not be reliable. You may have a situation where the only other person who experienced or witnessed the crime is dead. So you may have a murder victim who is not in a position to testify about anything. But you have a lot of evidence. You have a lot of biological evidence. You have, you know, you may have blood evidence, DNA evidence. You may have items that were found at the scene of the crime. You may have trace evidence that's found on some of the items at the scene of the crime and so on. And all of that needs to be tested, and it needs to be put together in a coherent way where it tells a story to the court. Because remember, if you had a witness, if the witness wasn't dead, if the victim wasn't dead, then that victim would be able to stand in court and say, I was walking down the street and the accused person then hit me on the head with a beer bottle. He dragged me into the bushes. He then raped me. And when he sobered up and realized I could identify him, he pulled out a knife and stabbed me in the neck, except that the victim is now dead. So the victim can't tell that story. But the evidence at the scene of the crime has got to be analyzed so that it can tell the story. Okay, so the body of the person will be, of the victim, will be examined by the pathologist. Okay. The pathologist would identify the stab wound, which caused exsanguination and then death of the victim. So the victim bled to death. The pathologist could potentially identify the fact that there's a large bruise or a lump on the back of the head, indicative of blunt force trauma. So the person was struck from behind. There's abrasions, let's say, from the person being dragged, that are consistent with being dragged from one area to the other. There's defense wounds on the arms of the victim, showing that she was struggling for her life. There's trauma to the sexual organs, indicating that there was a violent rape that happened and so on. So that's what you're going to see from the body. Then the examination of the crime scene itself, you're going to find the beer bottle, for example, which has the fingerprints and DNA of the accused person, but maybe also blood and hair from when the bottle was used to strike the victim. That's found sort of at the scene of the crime. You're going to maybe find drag marks and maybe find a shoe that's in the general vicinity. You're going to find blood that pooled at the scene of the crime where the person bled out. You're going to find maybe arterial spatter somewhere else that indicated she was stabbed in a certain place and then dragged off under the bushes, where the blood pooled under her when she died. So you're going to have the forensic experts that are going to together, because you're going to have a lot of them, working together, as in the case that I've indicated, who are going to be able to tell the story on behalf of the victim who can't because she's dead. So the role that you play as a forensic expert in the criminal justice system is huge, because to use the example that I gave you, the victim is not there anymore. The victim has been silenced forever in a very violent way. And where we would normally want the victim to testify about what happened to her, the victim's clearly not in a position to do it this way. So as a forensic scientist, you are going to analyze the evidence together with everybody else who's an expert in their particular field. And together you are going to, in court, come together and tell the story of what happened to the victim. So you are going to be, your role is basically to be the voice of a victim in court proceedings where the victim is not able to do so. So what's going to happen is that you would have examined the evidence, have analyzed the evidence, and each of you is going to tell your little piece of the evidence. That is all going to be put together by the prosecutors, by the lawyers that are acting for the parties and so on. And in so doing, the judge is going to hear the bits and pieces that you put together, okay, that comes together in the form of a story. You are going to, in your testimony, explain to the judge why this is the most likely scenario, or in your expert opinion, this is what happened, and it was the accused person that was responsible for making that happen. The judge is then going to look at all the evidence together. He's going to evaluate all the evidence, because remember, as experts, you are going to say, this, that, and the other happened. In my expert opinion, the accused did this, that, and the other, and that's what caused the death of this poor woman. Okay, the accused is going to say, no, not me, never, I was never there. But for example, his fingerprints may be found on the murder weapon. CCTV footage may show him in the general vicinity or walking away with the victim. So you have somebody who's an expert in analyzing those images that will be able to place him at the scene of the crime. You're going to have somebody who's done the DNA test, who's going to place his DNA on the body of the victim. You're going to have the blood expert who's going to say that there's a blood spatter that was found on his clothes that could only have landed there if he was standing right in front of the victim when she was stabbed in her neck. And the DNA of the blood found on him belongs to his victim. So in doing that, all of you as forensic experts are going to have a role in ensuring that justice is done for the victim in that particular matter. Yeah, there are cases where it goes a little bit pear-shaped. We had a very notorious case in South Africa some years ago as an indication of how a police investigation and the forensic evidence can sometimes be really messed up. This was a case where a young man was accused of murdering his girlfriend. So she was found dead in her apartment, and he was the suspect. The police bound relationship with this investigation start because the police just found a favor, didn't look at other potential suspects, and there were other potential suspects, but the police just focused on this on this boyfriend. And there was a wealth of forensic evidence that they found. So a footprint that was found in one of the bonnet. They were there was an ornament in the trunk. She'd been the victim had been bludgeoned to death. So obviously the hammer was something important. And you know, there were lots of little pieces of forensic evidence, but the police, and this is where if you're looking at things from an interdisciplinary point of view, especially in the criminal justice system, the police, the scientists, everybody has such a crucial role to play. So the evidence was collected at the scene of the crime. And the prosecution, because of the investigation by the police, placed a lot of emphasis on the fact that this bloody footprint was found in the bathroom. Fred, the boyfriend, said he hadn't seen his girlfriend from the night before, and she had been murdered much later than what he said he saw her. But the police tried to, and the prosecution tried to claim that it was his footprint that was in the blood. And if it was his footprint in the blood, then he must have been present either at the time that she was bludgeoned to death or sometime after, because that's the only way his shoe print could have been found in the blood. And the prosecution, the police officer who testified was adamant that the shoe print belonged to Fred, and he claimed that he had consulted with an eminent footprint or shoe print expert from the United States, who had confirmed that the shoe print belonged to a sneaker that belonged to Fred. Fred was very fortunate that his family had a little bit of money. So they were able to get their own experts, and they were able to fly this particular expert. His name was Bill Bodziak, and if you follow true crime and so on TV, you would have heard his name. And so they got him to fly to South Africa to come and testify in the proceedings. And what he said was that the prosecution was out and out lying about the fact that he said it was Fred's shoe print. What he said was that the prosecution had sent him photographs and so on, diagrams and so on of this shoe print, and asked him for his expert opinion on whether it could belong to Fred. And he stated categorically that he advised the prosecution that there was no way that it was from Fred's shoe. But the prosecution and their witness actually lied under oath in court proceedings. And you know, he was there then, and he offered his expert testimony as to why it could not have been Fred's shoe in that situation. So, you know, so you can imagine the judge was absolutely enraged at the fact that there was this almost misconduct by the state witnesses, where they were blatantly lying about the outcomes of the testing of certain evidence. I told you that there was a lip print that was found on a wine glass, and in that situation, the expert looked at the shape of the prints that were found on the wine glass, on the CD cover, and that kind of thing. And what they concluded was that the prints were actually planted, because when they looked at the shape of the prints were taken off something that had a curved surface and then planted on something that had a flat surface. And so the ridge detail that was found in the prints was somewhat distorted because it was lifted off a surface that had a slight curvature and planted onto a surface that was very, very flat. So, you know, you had a distortion in the shape, and it was clear from the ridge detail that you see that there was that. You know, so that was something else that the judge was, that was other evidence that the prosecution relied on, that the judge threw out as being unreliable, because clearly it had been tampered with and it had been placed in that particular situation. You also had the prosecution, who had their experts conduct further tests to show that the ornamental hammer that was found in Fred's vehicle was the one that was used, and that was the murder weapon. So what they did was they had to conduct their experiments in order to ascertain whether the injuries found on Inga's body, on the dead girl's body, would have been or could have been inflicted by this ornamental hammer. So as they do in these kinds of tests, a pig's carcass was used, and the ornamental hammer was then tested. So they used the ornamental hammer to bludgeon the pig's head to see whether the same type of injuries that the girl's body had could have been produced by the ornamental hammer. Interestingly, the hammer broke on the first or second hit that they blow that they struck to the pig's body. So what they went and did after that is that they took another hammer, a very, very different kind of hammer, and bludgeoned the poor pig's head. It was dead, but they tried to strike as many blows as Inga had suffered with this other hammer. And remember, I said at the beginning that when you have to show this link between each of the items in court in order for you to prove the thing from start to end. So clearly the police and the prosecution, the police's experts, the prosecution's experts, their experiment was flawed. Because first of all, the hammer was so flimsy that it broke on the first or second blow. And what the prosecution was trying to say was that this was the hammer that was used to inflict these multiple blows on the poor victim's head. And clearly it could not have been, because it broke so quickly. That there was no blood evidence or hair or anything that was found on the hammer. Just Fred's fingerprints were found on the hammer. But they decided that this was the thing that was used to bludgeon the girl to death. And obviously the court threw out the forensic evidence of the injuries and the number of blows, because the judge pointed out, and correctly so, that you can't just randomly take any hammer and try and reproduce the injuries and then say it was the one that was found in the accused's possession, because that doesn't make sense. It doesn't make legal sense, and it doesn't make common sense, because if you're trying to tie a specific piece of or a specific item to having inflicted those particular injuries, then you must be able to show in law that link between the accused person, the murder weapon, the victim, and clearly you had no link here between the accused person, the murder weapon, and the victim, because you had his fingerprint on the hammer, but his fingerprint could have gotten there. It was his. So obviously his fingerprints are going to be there. There was nothing tying that hammer to those injuries on that particular victim. So I think the point I'm trying to make here is that as a forensic scientist, you have got to understand something about the law and legal processes as well, because your expertise in the court case is so crucial, because it could be the thing that convicts somebody of fairly heinous crimes. And so you don't want to be the person whose testimony failed to secure a conviction. I think that is why it's so important for forensic scientists to actually be aware of the legal processes and the burdens that are placed on people giving evidence, and the duties that they have with regard to not misleading the court, to making sure that the interests of justice is served, rather than serving the interests of one party. Very often we don't see it very much in South Africa. I probably not so much in India, but sometimes in the United States, you have people who are professional witnesses, where all they do is they testify in court proceedings as expert witnesses. And, what it is selling your testimony to the highest buyer. And I think that in your professions, those of you in the science world, it's so important that you maintain the integrity of your profession, because your professions are held in very, very high esteem. And that's why in the legal system, we want your expertise and we want your knowledge to assist us to answer the difficult questions that we need to address in order to solve the legal issues that find their way into court. I just quickly want to talk about another case that we had here in South Africa. A judge was found murdered in his hotel room, and the police entered the room, and they very quickly wrote it off as being a suicide. So the first officers on the scene got in there, concluded that it was suicide. When the family was informed that, look, your dad has passed away and he killed himself, family members were saying, no, he was never, because of his religious beliefs and so on, he would never have taken his own life. He was not the kind of person who would ever have contemplated suicide. You know, when the family were told about what the hotel room looked like, or what the flat looked like, they said, something is not right here. Further investigation revealed that he was having marital problems with his then wife, and things were not very good with them and so on. And they insisted, because I think he was a judge and he was high profile and that kind of thing, the police were then forced to investigate further. So, you know, the circumstances were very similar. I think last year I was stuck in Nepal for about 7 months during lockdown. So I followed the Sushant case that you guys in India quite well. It was news. And it was very similar to that kind of case, where it was said to be suicide at first glance, but then there were all kinds of other factors that started to come to the fore, where the police now had to investigate to see whether it was a case of suicide or whether there was foul play involved in that. So what happened in our case with the judge is that the police went and investigated further. And then they found out, you know, from interviewing people and looking at witnesses and family members and that kind of thing, one of the things they found was that there was a bottle of whiskey on the bedside table, and there was a glass on the bedside table and something else. And people who'd known the judge for a long time said there is no way he would have left things on the bedside table. If he had a drink, he would take his glass and go to and rinse it off and put it away. He'd never leave the bottle of whiskey on the bedside table, because he was so meticulous, and he was all you know, almost like OCD, where there wouldn't be things lying around and that kind of thing. Also, for some reason or the other, the judge was found dead in his bed with the blankets pulled over him, and he was wearing 2 pairs of tracksuits. Running pants, sweatshirt, and then another pair on top of that. And nobody could understand why, first of all, that you would get into bed wearing a jogging outfit, and why you would get into bed wearing 2 jogging outfits. Then they looked even further, they investigated some more, and they found that the air conditioning unit, the temperature was turned up like really high, and it was not making very much sense. Further investigations indicated that the wife was going to be divorced, or he was planning on divorcing his wife. And because of how they were married, she would lose out financially, because they were married out of community of property. She would lose out in a big way financially if she was divorced. So the prosecution charged her, the wife, and her lover. It then transpired that she had a lover. She was cheating on her husband at this time. They charged the both of them with murder. And she claimed that no, well, you know, he must have died on that because they were claiming the time of death based on the decomposition of the body and so on as being a particular date. And then the forensic scientists basically said that, you know what, yes, that is so if we look at the body. But if we take into consideration the way he was dressed and the fact that the air conditioning was turned up and so on, all of these factors would have accelerated decomposition. So he actually could have died at a much different time than what the initial findings after the autopsy were conducted. Because remember, I talked about the additional clothing and being covered up and the air conditioning and so on. So the prosecution alleged that what she'd done was that she and her lover, that they'd actually tried to fix it to throw out the time of death so that they could escape liability, so that they would be in the clear, because they made sure that at the time, if the forensic pathologists and the other experts hadn't looked at the surrounding factors of the additional clothes and the air conditioning and the state of the room and so on, they would have, just on examining the body, would have concluded a certain time of death. And she and the lover made sure that they were accounted for at that particular time. But it was in further investigation when the other experts and the other scientists came in and said, yes, based on decomposition, it's that. But these are the factors in the room with his clothing and so on that would have accelerated decomposition. And so that time of death that you have is not actually accurate, because it, based on the other factors, it was some other time. So she and the lover were convicted of murder. She appealed some last year, and conviction was overturned, and got very lucky because she was the mastermind behind movement. So I'm going to ask if you have any specific questions that you want to address to me. I've kept my talk very, very general because I didn't want it to be too much of a legal talk. I just wanted to show your role and why this interdisciplinary interaction between our two professions is so important, just in the interest of justice, to make sure that everyone understands everyone else's role in these. So there's a question that I see on the chat about, have you experienced any forensic malpractices? I think you find it. South Africa, we are not as advanced forensically, I think, as maybe the United States, the UK. We have state-of-the-art forensic sciences laboratory and so on, but we are short-staffed with regard to the experts and the manpower. So that is why we have DNA, for example, that it's extremely backlogged, why we have ballistics experts that are working so hard, because there are not enough of them to go around. The problem also, I think, in South Africa, as with a lot of jurisdictions, is that our forensic sciences laboratory is attached to the South African Police Service. So it seems to be very biased in favor of the prosecution, because, you have the police who are investigating, the Forensic Sciences Laboratory, which is part of the South African Police Services, which is doing the actual testing and so on. And then you have the prosecution, who is relying on the police investigation, and the forensics experts who are attached to the police, who are preparing the case against the accused person. It's very expensive in South Africa to independently hire an expert on your own. So the cost of hiring a forensic expert for an accused person, it's normally out of beyond the means of most of the general man in the street to be able to have access to a forensic expert. So very often in criminal cases, unless the accused is represented by legal aid, which is free legal services to people who can't afford legal services in South Africa, if the accused doesn't qualify for legal aid, and if he is not a man of means, if he does not have the finances to do so, then he is stuck without having his own expert. So that is seen as a little bit of an injustice. And as I explained to you in Fred's case, where if we had to rely only on the prosecution's experts, you can see how things go wrong. In that case, it was very, very fortunate that Fred's family had some money. But, in order to pay for his defense, they were rich farmers and they had to sell almost everything, all the property that they had, in order to be able to hire the expert. So I think you have that that kind of thing. My students always argue with me that the system is not just if both parties can't have access to the same experts. Like I said, we expect that the expert, whether he's attached to the forensic sciences laboratory or not, his function in court is to assist the court, no matter who is paying for his services. Unfortunately, in cases like the one that I spoke about, sometimes the experts are biased or make mistakes, and it can result in a miscarriage of justice. Is there anything else that you want to ask?
Yes, ma'am, there is one more question. Why is eyewitness testimony given any value if it is unreliable?
Okay. I did talk about that a little bit, but it's unreliable just in general because it comes from a very subjective point of view. So, for example, if you think about it, or one of the exercises I do with my students in my class, they see me for the first time in their third year of study. They haven't seen me before because I only lecture third and fourth year LLB students, right? And on the first day of the lecture, I walk in and I do my lecture, and sort of about 2 or 3 weeks later, I ask them to describe what I was wearing, to say what they thought of me and this and that. And if I ask the question to 10 people, I get 10 different answers. And that I use as an indicator to show why eyewitness testimony is notoriously unreliable. First of all, 3 weeks later, they can't remember what I was wearing. And sometimes a matter goes to court a year later, where what a person was wearing whether the person was wearing high heels or flats, or whether the person was wearing pants or a dress, it may be important, because if one eyewitness says that the perpetrator was wearing a dress and CCTV footage shows the person who was leaving the scene of the crime wearing pants, then it's problematic there, because you've got two different pieces of evidence at odds with each other. So if you're describing appearance, I'm a short person. I'm only 1.6 meters tall. But to a person who's shorter than me, I'd be tall. To a person who's 1.8 meters tall, I'd be short. Or to somebody who's 20 years old, I'm old. So they describe me as an older woman. But to someone who's 70 years old, I'd be young. So subjectivity sometimes makes it unreliable. And memory, things like you forget details after a little while. You're open to suggestibility. So you saw a certain thing, and then you hear that other person, somebody else talking about the fact that, let's say you saw somebody who was wearing a black dress, and you listen to two other people arguing about the fact that whether it was a black dress or whether it was black pants, and you may become, oh, maybe I made a mistake, maybe it wasn't a black dress, maybe it was black pants, and then that's what you testify, because your mind has wrapped itself around that. Eyewitness testimony is so much more reliable if you have other testimony to corroborate it. So if your eyewitness is saying that I saw Desiree David at the scene of the crime, and she was walking away, and she was carrying a red handbag with her at that time, and you have the red handbag that is found at the scene of the crime with my ID in it, and you have CCTV footage. Okay, so I said, well, somebody stole my handbag and put it at the scene of the crime. Plausible. But then you have CCTV footage of me walking to the crime scene with my bag and walking away from the crime scene without my bag. So my story that somebody stole it from me is not plausible. So you have the eyewitness who said they saw me walking to the crime scene with a red bag. You have the red bag that's found there, that's corroborating the fact that I was there. You have the CCTV footage that's showing me with the red bag. So that eyewitness testimony is strengthened by the fact that there is this other evidence that is available. But eyewitness testimony on its own may sometimes be contradicted by the other evidence, and that is then what will make it unreliable in that situation. So although I said it's sometimes seen as unreliable, it's perfectly fine if you have other evidence that is corroborating, that is strengthening the eyewitness testimony. And of course, if you have eyewitness testimony and no other evidence, the judge in that matter is forced or must make a decision based on the evidence that has been presented in court. So if all you have is the eyewitness testimony and no other testimony that shows it to be reliable or unreliable, in other words, you've got no other testimony but the eyewitness testimony, it's not questioned, it's not shown as unreliable or incorrect or whatever, then it is the best evidence in that matter. It's the most reliable evidence. If there's nothing contradicting it, then the court, the presiding officer, the judge, will place much value on it. So it will be a very valuable piece of evidence, and it may be the thing that causes the judge to find the accused guilty. So it just depends on what other evidence is presented in that matter. All right.
Thank you so much, ma'am, for enlightening us with your knowledge. I now request Ms. Kajol Bhati to facilitate our guest.
For this knowledgeable lecture. It cleared numerous doubts related to the justice system and the role of evidence in law. Thank you so much, ma'am. Please accept this token of gratitude from our side.
Thank you very, very much for having me and for giving me an opportunity to share my thoughts with you. I hope that, it gave you a different perspective with regard to how to seeing your work as a forensics expert, forensic scientist.
Definitely great, ma'am. Thank you so much.
Thank you, ma'am. Thank you so much. Next speaker we have with us is Dr. PKS Nain, Dean, School of Mechanical Engineering, Galgotias University. Dr. PKS Nain did his BSc Engineering Mechanical from AMU, Aligarh in 1989. He got prestigious MTA Gold Medal for securing first rank in mechanical engineering. In 1992, he finished his ME with honor from IIT, Roorkee. During his ME, he was elected Chief Bhavan Counselor and Senate member in Roorkee University Student Association. In 2006, he was awarded PhD from IIT Kanpur. He got various scholarships and fellowships for his studies from only childhood till he finished his studies. The most prestigious was British Telecom UK Fellowship for his PhD work at IIT Kanpur. Dr. Nain started his teaching career from HBTI Kanpur. He has teaching and research experience of over 22 years in various institutes and universities. He has also attended several short courses which were organized in various parts of country by prestigious institutes. Dr. Nain's research interests are in field of soft computing, design and optimization. He has active research interaction in the field of optimization and evolutionary algorithms with IIT Kanpur, IIT Roorkee, IIT Guwahati, and IARI New Delhi. He works in genetic algorithms and has experience of over a decade in this area. He has published several international papers. He also got Best Paper Award at ICSCI 2005. He has also been session chair in international conference at SEAL 2010. He is currently reviewer of various international journals of repute like IEEE Transaction on Evolutionary Computation, Elsevier International Journal of Applied Soft Computing, Taylor and Francis International Journal of Green Energy, Elsevier Journal of Manufacturing Processes. Dr. Nain has successfully established distributed computation lab on AICTE funding of rupees 15 lakhs. He has been member of Board of Studies for Mechanical Engineering for UPTU, Lucknow, and has several times invited to teach the summer PG courses on optimization and CAD by various institutes of repute affiliated to UPTU, Lucknow. Dr. Nain is also member of several professional organizations like ASME and ICTE. Sir, I request you to kindly take over.
Thank you, ma'am. Thank you for your kind introduction. Am I audible now?
Yes, sir, you are audible.
Okay, so since we are slightly delayed, so I'll share my screen. So okay, is it visible to you all?
Yes, sir, it is visible.
Okay, so in order to reduce the bandwidth load, I will turn off my video for the time being. Okay, so my talk is today on vehicle accident reconstruction using energy analysis. So I'm trying to correlate the mechanical engineering into the field of forensic science. So what we are supposed to do in that and how we can give the reliable, extract the reliable piece of information which may be useful in making decisions about the recreating the scene of accident or the intentional accident or unintentional accident, and that has to decide who was doing wrong. So that is the objective of this today's talk. So the outline of the today's presentation is that first I will give the general introduction, what we are supposed to do as an engineer in the forensic science, and what are the weakness of other methods. When I am talking of the energy methods, there are some other methods also in mechanical engineering, like mechanics and dynamics issues. So that we will see what are the weakness in that method and what are the advantage of energy methods, which is the focus of the talk today, and how some details about how the energy method can be very easily applied to get the piece of evidence which are required by the court. And lastly, what are the new challenges which are coming in this field? So first we will talk of the vehicular accident, in which generally the engineers play an important reconstruction role. So automobile accident actually happen frequently, and due to these accidents, some litigations arise for the claim of medical expenses or disablement claims or the damage of property. So then we have to find out, okay, what has gone wrong during the accident. So accident reconstruction is a usual term used to describe the application of scientific principle, mostly the principles of physics and mechanics, and methodology to determine what has happened actually, the accident, and what was the possible sequence of events which happened just before accident which led to the accident. So the question can be whether the person who has hit somebody was over speeding or whether he was driving within the speed limit or that, because court will be interested in finding out those things. We have to generally find out the velocities of involved vehicle, the direction of their travel, and what at the time of accident, whether we were running at constant speed, we were decelerating or accelerating, then what is the sequence of event of accident which matches give the possible explanation of the causes of accident, and what are the forces which have come on the drivers or the concerned person who has suffered from the the accident, for disablement claims. So those things we have to check. So basically, what are the piece of information we have? The piece of information which we have are generally based on the physical evidences which are found at the scene of accident, otherwise which are directly related to the accident. So the scientific principles that are applied to analyze the accident are generally based on the physics, and they require us to calculate the various parameters within, not with certainty, but within a lower bound and upper bound, that what is the possible minimum speed or possible maximum speed at which the driver was traveling in the car at that particular time. So what was the probable distance at before which he started braking? So those things, reaction time, response time, all things we have to take into account. And finally, as we say, the statement or testimony of individual, those who were available at the scene of the accident, they are definitely taken into consideration, and the important clues can be obtained from them to relate the cause of accident. But as there was a question in the last portion of the talk, in the previous talk, a proper scientific analysis should be based upon verifiable facts and established natural laws rather than biased opinion of an interested party. Like, I made a mistake. I know in accident I was a driver and I knew that I have committed a mistake. I will always try to defend myself. I'll say I was very much within the speed limits, though this may not be the case actually. So we have not just the evidences should be verifiable by scientific analysis rather than my personal narration or narration of involved parties. Now, let me give an example that what is expected from an engineer in a case. Suppose we have a accident case in which there are three persons involved. First is Mr. John, who is traveling and who is the accused of rash driving. Okay, so he has hit another car of Mr. Wilson. So Mr. Wilson was driving perfectly, and his car was hit by Mr. John, who is allegedly was over speeding and has jumped a red light. Then we have a third person in our scene Mr. Smith, who is a witness who was driving behind Mr. John. So these three persons were there. So let us see what is the narration I have got from my reports. So the first narration which we got from our report was that Mr. John's car was traveling north in an intersection. So it was a crossroad and Mr. John was traveling north. And when it struck along a right side by a car of Mr. Wilson, who was traveling west in the same intersection. So one was traveling north and the second person was traveling west. So Mr. Wilson indicated that his light was green. The second person, who is not accused but who is the sufferer of the accident, he is saying that my light was green. And Mr. John also said that when I entered the intersection, the light was just amber, yellow. So he has right to travel. And that means Mr. Wilson actually has started early before his light turned green, and he was also driving fast. So that's why collision took place while he was correct. Mr. John was correct. So both the parties, Mr. John and Wilson, are saying that they are on the right side of the law. Now let us see, now comes the witness which is going to give us some useful information that Mr. Smith was a witness who was driving behind Mr. John from previous cross section also, and he said at the previous crossroad, Mr. John was trying to speed and so that he can reach to the next red light or next cross section before his light is turned red. So he was trying to speed and he was driving at least at the around 70 miles per an hour here, and that's he is indicating that Mr. Smith is basically on the wrong side. Mr. John is on the wrong side. So Mr. Smith, the witness, again said that Mr. John was ran into a red light at the next intersection and hit the other car. So Mr. Smith reportedly saw Mr. John's car at the intersection, and he is testifying that it was a red light. And Mr. Smith was saying that I myself was traveling at around 35 miles per hour, and that's why I am sure that he was approximately double my speed. So he must be around somewhere at 70 miles per hour. Now, what an engineer is supposed to do? In order to solve the accident, he will have to gather some information from the scene. And what that information would be? That information would be that distance from the sign is to be noted by Mr. Smith to the second intersection, and the distance from first intersection to second intersection and their lane weights, these things are to be observed and recorded. The time duration when the light turns green at the first intersection to when the light turns amber and then red on the second intersection, that time is to be also found out, so that we know that how much time is required to reach from one crossroad, one intersection to the next intersection. And we have also to consider that what is the make of Mr. John's car and Mr. Wilson's car, so that we know how fast they can accelerate and how fast they can reach to the claimed 70 miles per hour, how many seconds it will take. Second thing, the last and most important thing or piece of information would be the tire marks which will be there while braking on the second intersection, indicating the side slip of Mr. John's car after impact with the Mr. Wilson's car. And from the mechanics, we know that this will provide a very useful information to us as an engineer. So what I am supposed to tell after analyzing all these things? So I have to calculate as an engineer that whether Mr. John's car could reach the speed of 70 miles per hour as suggested by Mr. Smith, our witness, or whether Mr. John could reach the red light before it turned amber or red for the next cross section, at what speed Mr. Wilson would have struck Mr. John's car, and at what speed Mr. John's car was traveling when it was struck by Mr. Wilson. So I have, as an engineer, I have to make calculation and find out find out these values which matches the data which I have collected. So this is the job of an forensic engineer, which he is supposed to find out and answer in the courts. So in short, we have to find out the best theory and the best calculation and find out what fits to the best explanation for the current accident. So the accident reconstruction combines elements of disciplines of kinematics as an engineer and dynamics, material mechanics, and energy methods, human factor engineering, automotive engineering, and traffic engineering. So all these things we will have to, as an engineer, we will have to consider. So let us see what are the mechanics rules which we know from the 12th standard science, that what are the rules which will be applicable for a general, if I apply mechanics, not energy methods. Then one of the situation is, say, consider my car has fallen from a hill. So since it is a fall, so it may happen when vehicle runs off the cliff or bridge. So I can measure the height of vehicle fall, and the horizontal distance which it has traveled before impact on the ground. And it is very easy for me to calculate the velocity of vehicle with the help of measuring only two things. What are those two things? These two things are, I have to measure the distance h, horizontal distance h, and the height of the fall, the h, and the horizontal distance d, where it has hit the ground. So from that I can calculate the time, because since the vertical velocity at the initial point of fall was 0, so it is a gravity free fall if the velocity, the vertical velocity is concerned. So the time taken would be nothing but under root 2h by g, and the Vy would be g into t, and Vx, the horizontal component of velocity, would be the horizontal distance it has covered before hitting the ground, and the time we have calculated from the first equation. And the final velocity with which it will hit will be a vector sum of the Vx and Vy. If we take the second norm of the velocity vectors, then we will get the net velocity with which it has hit the car has fallen the ground. So it was a simple accident case. With mechanics information we can easily find out, but slowly the case will start to complicate. Now second case would be, if it is vehicle is in motion and a certain amount of energy is associated with it, and say on a level road, so it has a speed, it has a mass, so the half mv square, the mass into velocity square into 0.5, will be the kinetic energy associated with it. And when a body is stopped, so this energy has to be dissipated somewhere, and where it will go, we will have to investigate. And based on the energy investigation, that what were the possible consumers of the energy before the accident took place, and the vehicle from a certain velocity v to comes to the rest, so that we will that will help us decide the speed of vehicle which was the initial velocity of vehicle before accident, with quite a good accuracy. So vehicle is rest at the final stage, but initially it is traveling with a velocity with kinetic energy half mv square. And energy method are going to take over here, where they will take advantage of kinetic energy which is stored. So I am not directly measuring the velocity, but I am looking at the energy consumers, that where energy has gone. So the energy might be that when I am about to have the accident, I will brake hard. So there would be skidding marks of the vehicle tires. So if the vehicle tires, I can measure the skid marks and I can find out the distance of the braking and the kinetic characteristics of the vehicle. And I can find out how much energy it might have dissipated, knowing the friction value, knowing the weight of the vehicle. We can, as an engineer, we can find out it with a few calculations. So under normal circumstances, we have to assume that what is the function, whether the brakes were functioning properly or not, and how much vehicle brakes can convert kinetic energy into vehicle, and how much frictional work it might have done before in braking and skidding, and how much portion of energy have been dissipated by brakes and the mechanical brakes of the tire of the vehicle. So it will be distributed in two parts. One would be the energy dissipated in rubbing the ground by tires, which is leaving the skidding marks. And second is the brake is in touch with the brake drum, and it is trying to provide mechanical friction to the revolving tire, and it is trying to stop there. So how much energy will be dissipated? So basically, the kinetic energy will be dissipated in skidding and sliding, rollover and flips. Sometimes it may happen that the car rolls over and it flips also. So how much energy is required and consumed in rollover and flip, and the impact with other vehicle, in the previous case, it has hit some other car, so some part of energy also goes away, absorbed by the other slow moving vehicle, and the impact with the stationary roadside object, like you have the safety rails at the highways. So those aluminum rails are very soft, so that it doesn't damage much to your car, but they deform easily, so that they absorb certain energy, kinetic energy from your car. So they are broken, but during the braking scene, they absorb, the side rails absorb considerable amount of energy and slows your car effectively. And how much energy you have done in braking, and if you are going uphill, which is rare the case, but we have to consider, if we are going uphill, the potential energy of your car will increase, and that will slow it down. So that will also consume some energy. So the kinetic energy can go to the various forms which I have listed and just described out here. So that we have to see which portion is present in this equation, or which component is not present out of this whole mathematical equation. Now question would be with me, how I have to estimate these energies which has been consumed here. So the kinetic energy terms of calculation is complicated when we are having multiple car accidents, like we are having here in the expressways. When the car is hit, generally 2-3 cars accident in a quick succession, and you have to see that also. But in that, the kinetic, how much damage is done by first impact, second impact, which happen in quick succession. So there the calculation will be tricky and difficult. and your answer will have certain uncertainty as an engineer. So the initial velocity of vehicle is also important, but in these calculation, intermediate velocity at various points after the first hit and second hit, we have to be sure about them, or we have to have a reasonable guess which matches with the piece of evidence. So that has required. That is slightly difficult and depends upon experience. So these are the basically this equation. Those who are with the science background will understand it easily, that if a vehicle is there and it is having a weight and friction factor F, so weight of the or the normal reaction or weight of the body multiplied by the mu or friction factor of the surface in contact, that would be the force, which is the friction force which is coming on the tire. And how much distance, force into distance is nothing but energy. So F times W is the force and D is the distance to which the tire has skidded. So in this skidding, it has done work WD into F. So that calculates the energy dissipated in skidding. And you see, only by measuring the distance of skid distance D, knowing the standard values of F and the weight of the vehicle, we can find out how much energy has been dissipated in skidding while braking. We can, from this energy, half mv square, we can calculate, equate it to es and calculate the velocity as under root 2g df. That is a simple derivation. Or in case we want to say that I know the exact velocity of the vehicle and I want to find out the distance in which it will stop if I brake hard, then I can use the last formula, d equal to v square over 2gf, where d is the skidding distance. So this is based on the simple mechanics. But let us come back to our focus, that what is the crash energy dissipation, because we are going to talk about the energy method. So the impact may be in the front of vehicle, it may be on the surrounding sheet metal structure. So whenever a vehicle is hitting, the vehicle body is crushed. You see, if it is hitting from the front bonnet of the car is crushed and it is compressed. What is happening actually there? The work, the energy, kinetic energy is being dissipated in doing the work of the folding sheet metal, and that is slowing down the impact, which is being transferred from the hitting vehicle to the driver, because the ultimately driver safety is important. So you knowing the how much is the impact, how much is the crushing, how much is the length shortened by of the bonnet in crushing and folding, you can find out how much is the crash energy dissipated in the front accident. Similarly, greater is the speed of impact, the greater would be the depth of crush of the front crush of the car. So a depth of crush after impact damage is one of the very reliable method of calculating the energy absorbed by the vehicle. And the energy absorbed is solely kinetic energy. So that is half mv square equal to, if you equate these two things, you can easily find out what is the velocity at the time of hitting the car bonnet. So that velocity we can find out. And but it never happens. It happens that the impact happens after the skidding of the car. So we have to account for both kinds of energy. So we will have to account multiple sources of the energy consumption. What can be useful? As the ma'am in the previous talk was telling, that sometimes it happens that when the accident happens and when you go to testify into court, then there is a significant time for because of the procedural delays in filing the case and then filing the charge sheet, and then it is going to trial court. So by that time, memory fading effect of the all the witnesses goes, is a natural phenomena. So we cannot be sure of the narrations which we have got. So mostly the forensic scientists are sometimes asked to reach the spot after the vehicles have been moved from there. So he has to rely on the, but before doing that, good photographs are generally taken before the car is moved from the site. So good photographs of taking damaged vehicle and the surrounding area is a very important piece of information which a forensic engineer will be using. And with the help of these photographs, we can create the accident reconstruction easily. So where from I will find such information? For the, because the police is being trained to preserve this basic information. So generally in the investigation report of the police officer, you will find some informations about what is the good set of photographs they keep on the accident scene, what are the make, what is the dimensional information they measure, they keep a record in the charge sheet. So that a piece of information along with photograph provides a very important information to the forensic engineer. And then with this piece of information, we have to draft or construct 2-point perspective drawing and a sort of reverse method, that whether this much damage can happen by the sequence of event which is recorded in the charge sheet. And if they are matching, then your job is done. Otherwise, you have to express your opinion. What is your opinion and why you differ from the police theory? Then the takeaway is that whenever you are doing an energy absorption method, you have to see the crash depth, which may vary from vehicle to vehicle. Why? I'll give you example. If a vehicle, Maruti vehicle crashes, its bonnet gets crushed very or folded very easily in very less impact. But if you go to newer class of vehicles like Tata Nexon, who are very rigorous safety standards and the modern designs, then you will find there the safety is very much, and it depends upon the type of vehicle which you are using, that how much will be the crash depth on a particular velocity. The velocity may be 50 kilometers per hour at the time of hitting the car, but the damage which will be done in the Maruti car or the damage which will be done in a SUV, Fortuner SUV, will be very different because the crash depth will be different. So we have to see what kind of class of car was being driven and how much is the crash. So model is also to be considered. The same kind of analysis is applied to the rear end of the vehicle and side of the vehicle. So this is means reconstruction can be a very difficult task, and sometimes the actual accident scene is very far away from the engineer. So by the time forensic engineer reaches the site, the scene of investigation might have changed. So we have to find out, often rely between the conflicting information provided by witness and the accident participant, which can be actually confused or sometimes may be misleading intentionally. So participant may knowingly or unknowingly will lie about the events, and that has to be taken at the last. Under these circumstances, obtaining the factual information about the work to be with which the work is to be done can be very trying and difficult for a forensic engineer. So what is the piece of information which is most reliable for us? For us, the most information which is available in police accident report, that is the most important information. And second most important information is the photographs of damaged vehicles. The police accident reports will be basically recording location of skid marks, point of impact, and general layout of the scene, weather conditions, and the travel pattern of vehicle before the accident. Similarly, the photographs of damaged vehicle will can be very easily retrieved from the insurance company and the surveyors which are involved in evaluating insurance compensation to the accident victims. So the conclusion to the reconstruction part is that engineer may be asked in the end to provide information or opinions about many aspects of the case, including some of that are not related to the mechanical collision events. However, the engineer is nearly always asked to determine initial velocity of the vehicle before accident, and that he has to tell with certainty using the scientific analysis. So what are the limitation of some older techniques, like in which we said the how much is the time of fall, what is the velocity vector? These are the older mechanics technique. So there are three kind of techniques. One is the kinematic technique, other one is the dynamic technique, and the momentum method. These three basic physics principles can be used by the traditional analyst. So based on the kinematic analysis, it is very difficult because this technique requires the knowledge of velocity, acceleration, and distances involved for the vehicle from one point to other point. Point to point accident details are required, and we have to determine the various velocity vector, acceleration vectors at various locations, and which is very difficult to reconstruct. So kinematic technique is very hard to apply. Then we further improve it. We have to use a good guess, and we see with the iterative process that whether our theory is matching it or not. Then next is the conservation of momentum technique, in which the assessment of accident sequence often adds considerable equation. We know as a physics student, the moment is basically conserved. So the velocity and the angle at which the bodies, the two bodies hit and move in the direction after collision is basically can be analyzed with the help of the conservation of momentum techniques, but this also is a very tedious job. Then is the next is the energy method, and in the energy method basically we have certain advantages, and that's why we prefer to use energy method. In general, energy method don't require detailed point to point interior analysis of entire accident sequence. Rather, the energy method simply account for energy required to create all the damages and effects observed which are result of the accident. So here is a very important thing for a forensic engineer to note. The engineer is doing a post analysis. He's not interested in finding out what happened prior to it, but he's relying mostly on the damage effects which are recorded, which are always there, which will not go away with time or fade away with time. If the car is crushed in the bonnet, it is crushed and it is easily measurable, and it can be established even after 1 month before the repair takes place, of course. So the amount of damage that will provide me, I will see the various sources of energy consumed and the damage effects which has been done on the car, which damage effect which has been done on the side rails, and the skidding marks. All these are going to whether my car has flipped or my car has rolled sideways. That we have to see, and we have to estimate how much energy might have consumed in these things. So these methods are very simple, and this is nothing but application of first law of thermodynamics, which is nothing but conservation of energy, which says the energy of closed system remains constant. Energy can neither be created nor can be destroyed. So it can change form. So what happened? The kinetic energy changed its form into the various energies. And what are those various energy? Briefly, we have already talked, we will do it again. So, but in dynamics, basically, and momentum and kinematic evaluation, we require to make free body diagrams, we need to use vector algebra, we need point-to-point evaluation, which is very tedious. While the scalar method, the energy method is basically a scalar method and simply requires the accident and results. Not what was happening before is not required. So energy method is an excellent technique for accident reconstruction, and all the basic information necessary to apply the energy method is normally contained in the police report and the surveyor's photographs. So let us see what is the first fundamental principle. The fundamental principle is that in the reconstruction of vehicle accident, the application of first law may not be readily apparent. So we have to use a corollary, and that corollary says that work done on a particle by a force is equal to change in kinetic energy of the particle. Work done is the energy form, and that is equal to force into distance, resisting force into distance, and that should be equal to the kinetic energy of the particle, and particle in this case has to be a big particle, which is a car. For mechanics point of view, it is a particle. So in the broad terms, it means that the initial kinetic energy of vehicle will be equal to the work that the vehicle does while it is coming to stop after accident. So this includes the work necessary to skid, change elevation, roll over, or crush side protector metals of the highways. So all these things we have to consider. So in short, what can be involved in a vehicle accident? The general fundamental equation would be the kinetic energy is equal to the energy consumed in crushing of the bonnet, crushing of the side of doors, the crushing of at the back end, whatever damage is there. So all the energy will be consumed that we have to take into account. Then the energy consumed by the skid friction when we are braking and the skid marks were coming on the road, that we have to take into account. And if the car has rolled over or flipped, then the energy consumed in rolling over is also to be considered. And the energy change due to elevation, if we are going uphill, we are going on a level road, or we are going downhill, that the change in potential energy is also to be accounted. And other type of energy consumption like sound, heat, because while braking, the mechanical brakes of the vehicle generally turn it into a heat energy. The kinetic energy is turned into the heat energy. The noise which is being generated at the time of accident is nothing but sound energy that has to be taken into account. But this component is generally very small, 1 to 2%. Now let us see what is kinetic energy and how we can model them. Kinetic energy is half mv square, where m is the mass of the vehicle, which is weight divided by gravity, acceleration due to gravity, and v may be the initial velocity. If there is a partial accident, then the change in kinetic energy before the event and after the event would be the energy consumed in the accident. If we are talking about the frictional energy, basically we know the frictional energy of a block which is kept at the surface. So the mu is the friction coefficient, n is the normal reaction which is equal to weight of the body. So mu we are representing here as a friction factor, w is the weight of the car, and s is the distance through which you have measured the skidding mark on the road. So that has to be used, and then you have to equate kinetic energy to the frictional dissipated energy, and then you have to find out the initial velocity of the body. the car. But one important thing is, if the car is taking a turn, then what to do? If it is moving on a, while it is having the accident, it is moving on the curved path while braking, then how it will be measured? Then you have to measure the arc distance, not the displacement, means from initial point to final point. If you measure the straight line, the distance may be smaller because the car has really moved on a curvilinear path. So the skidding marks would be larger than this measurement, and actual skidding mark length is to be used in the calculation. In case all four tires of car travel different distance, then you have to measure the individual travel of all the car wheels and then take a mean value of all the four travels which you have measured. And then you have to equate it to kinetic energy, to the frictional energy. Then here the survey reports of various forensic books or handbooks is going to help you, because now you know if the car is traveling on a gravel and dirt road, since the friction coefficient depends upon the nature of surfaces in contact, you can use this table which I am showing here. That gravel and dirt road will give you friction of friction factor F of 0.35. If it is a wet grassy field, the friction would be less, but it would be 0.2 when you are braking. If it is dry asphalt, then it is 0.6. Wet, if it is raining, then it is, this value reduces to 0.45, and that's why the climatic condition at the time of accident are also important. In the snow covered road, it becomes more slippery and the friction coefficient reduce in the ice, and dry concrete it is higher, and wet concrete it is slightly low. So based on these values, these are available in the handbooks. You can use the value of F here. Weight of the car you know from the car manuals, and the skidding distance you have measured, and that you equate to half mv square. You can easily find out the velocity. Now further to change, what happens if the change in elevation happens? If you see this car is going to roll over in this direction. So if this point is the center of gravity of this car, center of mass rather, then B is the height from the ground and A is the distance about from the tire edge about which is going to turn. So if you see, what would be the net elevation change, H1 minus H2 would be nothing but under root A square B square minus B. And similarly, when the vehicle is going to flip, then it is hitting an obstruction in front, then definitely the initial height of the center of gravity was B, and when it flips over and goes to 90 degree, the height would be A. So the change in elevation would be A minus B as shown here. So with the help of mg into h, that is weight into the change in elevation, you can find out the energy involved in rolling over and flipping of the car. I hope I am clear. Now, when the bonnet is crushed or the car body parts are crushed, there are two parts of deformation as an engineer. One is the elastic region in which the deformation is recoverable, that when I remove the load, the deformation is recovered, and the portion of the enhanced portion which is after the elastic region, the non-linear region rather, that is the plastic deformation region, and whatever energy is absorbed here, that is not going to come back after the loading is removed. So when the bonnet is crushed or folded, the part of it is the elastic deformation and part of it is the plastic deformation. You might have seen, if you hit a very rigid object, your car after stopping will back slightly, 6 inches, 8 inches. So that happens because of the elastic nature of the deformation. Part of the deformation is in the elastic energy. And this from mechanics we know, this is the basically curve, stress strain, force deformation curve for the steel. But now we have the composite materials also being used in bonnets and others. So this can be significantly different. So we have to see the material with which we are working in the car. So elastic energy would be equal to half kx square, and the plastic energy, we take an average mean stiffness of the material, and k bar x, where x is the deformation or the damage which has been done in the uh, car, that has to be measured, and with the help of these values we can do that. So here I am showing you in the forensic manuals, basically instead of this value, they use Fc into X, which is they call Fc as the crush constant, and X is the deformation, which the distance through which the bonnet or the metal sheets of the car has been crushed. And this is in this table is in foot, pound, and seconds, because I have taken it from a foreign source, so European source. So we can also have a similar conversion factors, and we can find the average crush constant. And if you see, the crush constants are dependent upon the make of the car also, and the size of the car also. So in general, we will come back to our fundamental equation. In this, we found kinetic energy, which is half mv square, and then we have the consumed by crushing, energy consumed by crushing, and I have shown you how to find out with the help of crush coefficient. This is the energy dissipated in crushing. Then energy in the consumed in the screed friction, we have already found out, it is friction F into W into D. The energy consumed in rollover, we have studied the center of gravity displacement in rolling and flipping. By that method, you can model ER. The EE is the energy change due to elevation. If you are, it is in the hilly condition, then mg delta H is going to be this energy. And lastly is the other type of energy consumption, like sound, heat, which is generally very small, 1 to 2%. So what are the new challenges which are coming to a forensic engineer is that we are now having anti-locking brakes in all the modern cars. And you might have seen the skid marks of the anti-locking brakes are very difficult to find, because they are stopping only for few movements. So you will find the skid marks in the brakes, very small fraction of second the tire is locked. And the advantage of skid lock anti-locking brake is that skidding is avoided. And skidding, when skidding is avoided, basically effectivity of braking increases. Secondly, due to anti-locking braking of feature, the change of direction of the car while braking is possible, which was not possible in these older case. Hence, now we have to rely more on the manufacturer's data. So this is a new challenge which has come in the forensic engineering area. No or very faint skid marks are obtained, and if you go about measuring the skid marks after 2-3 days, it will be very difficult if the car was having anti-locking brakes. Next is the determination of crush constants for the composite materials which are recently used in body parts of the automobile manufacturers, in order to make them light, less costly, more rigid, more energy absorbing, so the energy dissipation is fast. So that data is required from the company manuals only, and company basically are doing lot of crush tests before launching the vehicle into the market. So we have to rely on their crush data as a forensic engineer. These are the new challenges. So the reference for the today's talk was the book by Randy and Introduction to Forensic Engineers. It has multiple things I have could only cover few points in given in it. And the motor vehicle accident reconstruction causes and analysis by Limpert. These two are the valuable reference for today's talk. And now the house is open for the questions, if any. Anybody who is interested in asking the question can ask the question now. Hello.
Okay, sir. Thank you so much for sharing your mastery with us. I think there is no more questions. Participant can ask their questions if they want.
So I hope this much is all which I can cover in this. If there is not any question, I think we can close.
Thank you so much, sir. I now request Ms. Kajol Bhati to felicitate our guest.
Thank you so much, sir, for your lecture on vehicular accident reconstruction using energy analysis. It was an informative session. Please accept this token of gratitude from our side.
Thank you very much, and thanks to the Department of Forensic Science for giving me opportunity to share this interesting view of an engineer on the forensic science. Thank you.
It was our pleasure, sir. Thank you so much.
Thank you so much, ma'am. Thank you so much, sir. Dr. Rakhi Agarwal couldn't make it today, so now we are ending our session. I, on behalf of everyone here, extend a very hearty vote of thanks to all speakers for gracing your important work and sharing with us your findings and opinions today. Feedback link will be shared via mail. Kindly fill it, context of getting attendance and certificates. And kindly join FDP day 3 tomorrow at 3:30 p.m. sharp. Thank you all for your cordially cooperation.